Hi, my name is Lambda. I'm a postdoc at Lure Pachter Lab at Caltech in Pasadena. So today, I would talk about the Voyager project, which brings the tradition of geospatial data analysis to spatial omics. And you can scan the QR code to look at the preprint to read more about this project. So with hundreds of institutions around the world using various types of spatial transcriptomics technology, spatial transcriptomics is already mainstream. The number of new publications in this field has risen drastically since 2019. However, there has already been decades of research in spatial data analysis prior to 2019, and mostly in geography. And this timeline shows some of the methods used in Voyager borrowed from the geospatial field. From the decades of research, we also have some well-established data structures and data analysis methods for different kinds of geospatial data. So if we can draw an analogy between geospatial data and spatial omics data, then we can borrow some of the existing tools from geospatial instead of reinventing the wheel. So this is a schematic of the Voyager project, and first I will begin with the spatial feature experiment data structure, which the Voyager project is based on. The heart of spatial feature experiment is simple features, which is a well-established and standard way to represent geometry, vector geometry data in geography. And in R, it can be accessed with the SF, the SF package. So basically, the geometries in our field, it can be the cell segmentation or nucleus segmentation polygons, and they are described with the coordinates of the vertices, or we can also have coordinates of cell centroids or transcript spots from SMFish. Behind SF, we have the GeoC++ library for geometric operations. So with these operations, we can find whether two geometries intersect, or if one is entirely within the other, or we can also find the exact geometry of the intersections, and so from here we can find which cells are in which Visium spot, how much they overlap, and which Visium spots or cells are in which pathologist annotated region. And we can also use the geometries to perform operations on the spatial feature experiment objects. This is a schematic of the SFE object. So it's based on existing data structures on bioconductor, spatial experiment, and single cell experiment. In single cell experiment, we have the gene count matrix, and we have a row and column metadata for the gene count matrix. So analogous to the row and column metadata, spatial feature experiment, or SFE, has SF data frames to represent the geometries corresponding to the dimensions. So it can be transcript spot points, or it also can be Visium spot polygons, or nuclear cell segmentations. And in addition, we have annotation geometries, which are geometries that are part of the data set, but they do not directly correspond to the dimensions of the gene count matrix. So these can be, say, histological regions or tissue boundaries. And also because some spatial data analysis methods require a spatial neighborhood graph, which basically show like who's the neighbor of whom. And so analogous to the geometries, we also have the graphs for rows and columns. So now we have a new data structure. What are we going to do with it? So basically, Voyager to SFE is just like a scalar to a single cell experiment, or Seurat to Seurat object. So Voyager performs exploratory data analysis to SFE objects. So basically, exploratory data analysis, or EDA, is like it's important to understand what you can do before you learn to measure how well you have simply done it. However, usually those methods do not take the spatial information into account. So we also have ESDA, or exploratory spatial data analysis. So that's EDA specifically for the spatial aspects of the data. So a very important aspect of ESDA is spatial autocorrelation. The spirit of that can be summarized in Tauber's first law of geography. Everything's related to everything else, but near things are more related than distant things. So we can have positive spatial autocorrelation, in which nearby things tend to be more similar than distant things. And example is weather. Negative spatial autocorrelation means that the nearby things tend to be more different than distant things. There aren't as many examples, but one of the obvious examples will be the chessboard, where the neighbors must be different. I'm definitely not the first person to bring the geospatial tradition to spatial omics, and this slide lists many existing methods that have already done so. However, these packages really tend to focus on very specific tasks, like how well you have simply done it, whereas the what you can do part remains unaddressed. And Voyager implements general ESDA to bridge this gap. And because there are many ESDA methods, we have implemented a systematic uniform user interface to minimize the learning curve. So back to the schematic, in the middle here, that's sort of a that's a summary of the uniform user interface. We have, in addition to visualizing gene expression in space, we have also implemented plotting functions to visualize the spatial analysis results. And all the palettes here are colorblind friendly. We have written a comprehensive documentation website, and you can scan the QR code here to look at the website. So that we have vignettes on the website using many popular commercial spatial transcriptomics, also like codex proteomics data sets. And to make sure that the documentation website's reproducible and scalable to larger data sets, the entire website is built automatically on GitHub Actions, and we also generated the Google Colab notebooks for you to play with the tutorials. So here I will focus on the R implementation on bioconductor. But if you prefer Python, then we also have a Python implementation of Voyager. And although that's only for like the univariate methods, we have written compatibility tests to make sure that the two implementations give the same results. So now we can get into some case studies. So the first one, it's even before spatial statistics. It's just based on geometric operations. It's from a mouse skeletal muscle data set from Visium, and so here on Visium spots I plot in counts where that's the library size, and we see that actually so with geometric operations, I can find the mean area of the myofibers that intersect each Visium spot, and so here we see that like there's correlation between the library size and between library size and myofiber area. In that, larger myofibers tend to have a smaller library size, so yeah, this means that if the library size is actually biologically relevant, and it might be problematic to treat it as a technical artifact that's commonly done. So next we can get into some spatial statistics. The most commonly used spatial statistics is Moran's I, which is a metric of spatial autocorrelation. So it's kind of like a Pearson correlation between the value at each location and the spatially lagged values, like the weighted average of the neighbors, although it's not exactly the same. And so Voyager not only implements parallel processing to compute Moran's I for thousands of genes, but also has based on the SPDEP package, the Monte Carlo simulation and for Moran's I, and so there are also other spatial autocorrelation metrics like GRC and global G and so on, which will review like different aspects of spatial autocorrelation from Moran's I. So Moran's I is only one number, and so that's one number for the entire data set, and that would obscure a lot of the local heterogeneities. So we also have local versions of those spatial autocorrelation metrics, such as local Moran's I or local GRC and JEDIS or GI star and so on. So basically, for local Moran's I, it's basically the contribution of each spot or each location to global Moran's I, and here we can, from local Moran's I, we can see like the positive values here are the more homogeneous regions, and the negative values are the more heterogeneous regions for this gene myosin heavy chain 2. So again, like for the local metrics, we can also perform Monte Carlo simulations, and also like get a there are also analytical p-values for. So those were those are just the univariate spatial statistics, but often we have hundreds, if not thousands, of genes in our data set, and nobody wants to manually check like one gene at a time. And so Voyager also supports also implements some multivariate spatial data analysis methods borrow from the geospatial tradition. So one example here is the multi-static PCA, which is a form of spatially informed PCA. In non-spatial PCA, we will try to so we'll try to maximize the variance explained by each PC. But in multi-static PCA, we will try to we will maximize the product of variance I and variance explained. So for instance, here, like in the mouse liver merfish dataset, it has single-cell resolution from non-spatial PCA. Like it takes, so the first two PCs do not seem to have much spatial structure, but and it takes until like PC5 to get to the hepatic zonation, which has spatial structure. In contrast with multi-static PCA, because we have the product of variance I and the variance explained, so the first PCs would need to not only explain more variance, which will often mean like biological relevance, but also have spatial structure for large positive variance I. So in first two PCs, we get to the hepatic zonations right away. But here, note that because the spatially weighted covariance matrix is not positive, it's not positive semi-definite, we can have negative eigenvalues, and here we do have a pretty significant negative eigenvalue here. And so this means this PC should not only explain more variance, but also it needs to have negative spatial autocorrelation. And turns out that the negative spatial autocorrelation is biologically relevant, and this PC will separate the endothelial cells and Kupffer cells from hepatocytes based on gene loadings. So these are only like a few examples from many other like case studies shown in the documentation website. But Voyager is still an ongoing project, and so for the next release on bioconductor, I'm in next April. So right now I'm still working on those. Like I'm still working on the better dealing with like ESDA in multiple samples. Also in three-dimensional data and on disk geometric operations for larger data sets. Finally, no analogy is perfect, and so there may be spatial phenomena that are only present in histological space, but not in geography, and those should be accounted for. And also, like I briefly mentioned the colorblind friendly palettes, and so we'll also like to extend this framework to be accessible to more kinds of disabilities beyond the colorblind friendly palettes. So I would thank the entire team that have contributed to this project, and this is an international team from not only at Caltech, but also in Iceland and Germany, and I would thank these funding sources. I can thank you, and I'll take questions.
Questions? I'll start with could you briefly, I mean, you mentioned this direction for 3D data. Could you briefly sort of give some indication of what would be involved, like in going from 2D to 3D? Like, would it be a straightforward generalization, or would it need like a lot of new, very different types of techniques?
I think maybe not necessarily. I think one challenge is that I think that what's special about spatial feature experiments, the geometric operations. However, like geometric operations, like it's with the geos backend, it only works for 2D, but not in 3D. So I have to like hack around that. But it's generally like a 3D geometric operations are much more challenging, and those are usually addressed in like the CGI, like a computer graphics tradition instead of the geospatial tradition. I think it's probably now the most urgent right now, because the vast majority of spatial transcriptomics datasets are like pretty much 2D. I think I think another challenge will be what shall we do with the z-planes, because actually I do know there's some 3D datasets like in Visium that would take like a many sections along the like 0 sections with various spacings, and but then in that case the resolution in the z direction would be very different from those in x and y direction, and I think those might have implications.